Hi and welcome. This lecture is going to focus on the plasma membrane. The structure of the plasma membrane and how it functions in the cell. The plasma membrane is a fluid mixture composed of equal parts of lipid and protein when you look at it by weight. Now, the function of the plasma membrane is to regulate the movement of most substances in and out of the cell. It is a very effective non-polar barrier to most substances. However, small and non-polar substances are able to penetrate without assistance. It contains several different types of lipids. That includes phospholipids, cholesterol, and glycolipids. The plasma membrane is said to be a selectively permeable membrane, and that is because of its ability to regulate the movement of these substances. Let's take a look at phospholipids first. Phospholipids are an amphipathic molecule, meaning that they have a polar head and nonpolar or hydrophobic tails. Most of the lipids in the membrane are phospholipids. They will form what's called a bilayer, or two parallel sheets of the molecules in which the tails are facing each other on the internal side, and the heads, or the polar heads of the phospholipids, are facing towards the extracellular fluid and the intracellular fluid. It is termed, when you look at them together, a phospholipid bilayer. It ensures that the ICF and the ECF remain separate. Now, the ECF can sometimes be called the interstitial fluid, or the fluid that you see among tissues. Now let's take a look at a second kind of lipid component. That is cholesterol. It is scattered within the phospholipid bilayer. Its function is to strengthen the membrane. It helps stabilize the membrane against temperature extremes. Temperature will affect the fluidity of the cell membranes. A third kind is glycolipids. Now, a glycolipid is a type of conjugated organic macromolecule. It is lipids with attached carbohydrate groups. It is located on the outer phospholipid region only, and it helps to form something called the glycocalyx, or the coating of sugar on the cell's surface. Here is a picture showing the parts of the plasma membrane. As you can see, if you look at the plasma membrane itself, you can see the individual phospholipids. That's every one of these molecules is a phospholipid. Now, the phospholipid has the polar head, that's the round circles here, and the nonpolar tails, which are on the inside. Now, the nonpolar tails laying next to each other allows the phospholipid bilayer to have that fluid-like component. It is basically like two oils rubbing against each other. Here is the cholesterol molecule. Keep in mind that that is there to help with strength. And each of these are proteins that you see within the phospholipid bilayer. Now, we are going to learn that there are different kinds of proteins that you can have. Here is your glycolipid, and this is the carbohydrate portion, and then you have the phospholipid, which is the lipid portion. Now, here is a glycoprotein. You can see the carbohydrate portion and then the protein portion here. When you look at the proteins within the plasma membrane, they are termed to be membrane proteins. They compose half of the plasma membrane by weight, and they have the ability to move about the fluid bilayer. They can float throughout it. Most of a membrane's functions are determined by the proteins that it has within it, and you can classify these proteins as either integral or peripheral proteins. When you look at the integral proteins, these are proteins that are embedded within and extend across the lipid bilayer. They have hydrophobic regions, which will interact with the hydrophobic interior, and they will have hydrophilic regions interacting with the hydrophilic regions of the bilayer. Keep in mind that whenever you are looking how compounds dissolve each other, this simple phrase, and that is like dissolves like. So hydrophobic will dissolve hydrophobic, and hydrophilic will dissolve hydrophilic. Often glycoproteins have the carbohydrate portion attached to it. Now, the peripheral proteins are not embedded in the lipid bilayer, and they attach loosely to the surfaces of the membrane itself. We can often take those kinds of integral or peripheral proteins and categorize them based on what their function is. One function is transport. You can have transport proteins that help regulate movement of substances across the membrane. Examples of these would be channels, carriers, and pumps. You can have cell surface receptors. These will bind ligand molecules released from a specific cell. Now keep in mind that the term ligand is a generic term that simply refers to any chemical that can bind to these surface receptors. They can also bind receptors on another cell. Example would be neurotransmitters and/or hormones. A third is identity markers. These are proteins that communicate to other cells. The example would be immune system cells that can distinguish healthy cells from foreign cells. We often call these antigens. When you look at the proteins within the membrane, a lot of those proteins are going to be enzymes. These are kinds of proteins that will speed up chemical reactions. So they are said to catalyze chemical reactions, and catalyze means to break down. Next, we have anchoring sites. These are sites that secure the cytoskeleton, or the skeleton that is on the inside of the cell, to the plasma membrane. We also have cell adhesion proteins. These are proteins that will help attach cell to cell. Here's a picture showing the kinds of proteins that you can find within the plasma proteins. Here is a transport protein, and we can see that this is a channel that is allowing the molecules to move through. Now keep in mind, only small nonpolar molecules can move through the plasma membrane itself. So these types of molecules are usually large, nonpolar, or polar molecules. Secondly, we have the receptor, and the receptor are binding to ligands that are floating within the ECF, or the extracellular fluid. Here is a identity marker. We can see that this is a glycoprotein. This is the carbohydrate portion here, and this is the protein portion. Here we have an enzyme. We are going to learn about enzymes later, but the substrate is the chemical that is being acted on by the enzyme. It will come into what's called the active site of the protein, and it is going to act on that chemical and then produce the product. Here we have an anchoring site. Now notice that this protein is anchored within the plasma membrane itself, and it is also attached to the cytoskeleton. This has helped giving the cell internal support. Lastly, the cell adhesion molecule. This is cell number 1, And here is cell number 2. Here is the 2 cell adhesion proteins. This is the interstitial space, so we can see that these proteins are attached to each other at this point, and they are helped to hold the cells together.